Okay, so welcome back to this next video in which we are discussing type 1 phosphoinositide 3-Kinase enzymes. Okay, so remember we discussed that all phosphoinositide 3-Kinase enzymes add a phosphate group onto the alcohol group that comes off the third carbon of the inositol ring within a phosphoinositide. How they differ and how we form these three different types is by having different substrates, basically. So type 1 phosphoinositide 3-Kinase enzymes work on phosphatidylinositol 4,5-bisphosphate and create phosphatidylinositol 3,4,5-trisphosphate. Type 2 phosphoinositide 3-Kinase enzymes work on phosphatidylinositol 4-phosphate and create phosphatidylinositol 3,4-bisphosphate. Finally, type 3 phosphoinositide 3-Kinase enzymes work on phosphatidylinositol molecules and create phosphatidylinositol 3-phosphate molecules. We are now going to completely focus in on type 1 phosphoinositide 3-Kinase enzymes. Okay, right. So firstly, we're going to talk about the structure of type 1 phosphoinositide 3-Kinase in a very basic sense, okay, and then we'll draw more accurate pictures, more in detail pictures later on, okay. So for now, let's start with a very basic picture of what type 1 phosphoinositide 3-Kinase enzymes look like. Okay, right. So basically, type 1 phosphoinositide 3-Kinase enzymes consist of two portions. Okay, so they consist of two proteins bound together. Okay, one of these proteins is what's known as the catalytic subunit of the type 1 phosphoinositide 3-Kinase. Okay, so this subunit that I have written C in, this represents the catalytic subunit of the phosphoinositide 3-Kinase enzyme of the type 1. Okay, and then the other subunit that we've got bound here, we'll put an R in this box. Okay, this is the regulatory subunit. Okay, so overall, the type 1 phosphoinositide 3-Kinase enzymes consist of these two separate subunits bound together, a catalytic subunit, which I'll highlight in blue here, and a regulatory subunit. Okay, now there is not just one catalytic subunit and there is not just one regulatory subunit. That would be the really simple thing if there was one type 1 phosphoinositide 3-Kinase catalytic subunit and just one type 1 phosphoinositide 3-Kinase regulatory subunit, but it's not the case. Instead, there are actually 4 genes that code for catalytic subunits of type 1 phosphoinositide 3-Kinase enzymes. Okay, and these 4 genes are known as the phosphoinositide 3-Kinase, which I'll just abbreviate to Pi3K. Okay, then we put C for catalytic subunit. Now, strictly speaking, of course, you'd like it if we could put a type 1 in front of there to tell us that we're dealing with a type 1 phosphoinositide 3-Kinase. Generally, people don't clarify that. Just as you can abbreviate PIP3, just as you can abbreviate Pi345P3 down to PIP3, and people will know what you're talking about. If you don't specify which type of phosphoinositide 3-Kinase enzyme you're working with, people will assume you mean type 1, okay, because it's the main form of phosphoinositide 3-Kinase. Okay, right, so phosphoinositide 3-Kinase, catalytic subunit, and then there's the alpha one, which is then given the letter A. Okay, then there's a beta one, okay, which is called Pi3K catalytic subunit B. Okay, B for beta. Then the next one is the Pi3-Kinase catalytic subunit, and then it's the gamma one, so we put a G there. And then finally, there's also the Pi3-Kinase catalytic subunit delta. Okay, so we'll put a del there. a D there, rather. Okay, right. Now, these are the names for the genes. So if you see someone referring to Pi3KCA, this refers to the gene rather than the actual protein. Now, all of them produce proteins that have a certain weight, usually. They have a weight of around 110 kilodaltons. Okay, and for this reason, the actual proteins that these genes produce are known as P110s. Okay, and P just means protein, and then it's of molecular weight 110 basically. Okay, and the protein which the Pi3-Ka gene produces is known as P110 alpha. The protein which the Pi3-Kb gene produces is known as P110 beta. Okay, the protein which the Pi3-Kcg produces is P110 gamma, and finally the protein which the Pi3-Kcd gene produces is known as P110 delta. Okay, so these are the 4 proteins which you can actually use as a catalytic subunit of a type 1 phosphoinositide 3-Kinase enzyme. Okay, so all of these proteins can be put in here as the catalytic subunit. Okay, rather 4 proteins rather than just 3. Okay, so basically the whole phosphoinositide 3-Kinase enzyme is now named after which catalytic subunit you have. Okay, so I don't care what regulatory subunit you have. The way I name the entire Pi3-Kinase enzyme is via the catalytic subunit. Okay, so if your Pi3-Kinase enzyme has as its catalytic subunit P110 alpha, then I will call that whole enzyme now a Pi3-Kinase alpha. Okay. Similarly, if your catalytic subunit is P110 beta, then the entire Pi3-Kinase enzyme will be called Pi3-Kinase beta. Okay, and so on. If your catalytic subunit is either P110 gamma or P110 delta, then the entire thing is then called a Pi3-Kinase gamma or a Pi3-Kinase delta. Okay, so we do not name the Pi3-Kinase according to which regulatory subunit you contain. We just look at what the catalytic subunit is, and we name the Pi3-Kinase enzyme according to which catalytic subunit it has. Okay, so that means that as far as our naming system is concerned, there are 4 different types of phosphoinositide 3-Kinase of the first type. Okay, so now type 1 phosphoinositide 3-Kinase enzymes are going to be broken down, okay, into even more classifications. Okay, so there's a further classification. We can classify the phosphoinositide 3-Kinase enzymes according to whether they are type 1A or type 1B. Okay, so the type 1A phosphoinositide 3-Kinases include the Pi3-Kinase alphas, the Pi3-Kinase betas, and the Pi3-Kinase deltas. Okay, so the phosphoinositide 3-Kinase enzymes which have as their catalytic subunit either P110 alpha, P110 beta, or P110 delta. Okay, so alpha, beta, or delta. Okay, so all of these Pi3-Kinase enzymes, they are all type 1A Pi3-Kinase enzymes. Okay, then that leaves the final type, the final Pi3-Kinase enzyme, Pi3-Kinase gamma, as a type 1B Pi3-Kinase enzyme. Now, what is the main difference then between type 1A Pi3-Kinase enzymes and type 1B Pi3-Kinase enzymes? Well, it's based on what activates them. Okay, the type 1A Pi3-Kinase enzymes are going to be activated by receptor tyrosine kinases. Okay, whereas the type 1B Pi3-Kinases, those are going to be activated by the beta-gamma subunits of heterotrimeric G proteins, G-beta-gamma. Okay, so they are regulated by very different things. We are going to focus on the type 1A Pi3-Kinase enzymes. Okay, we will look at the structure of type 1A Pi3-Kinase enzymes, and we will look at the regulatory subunits that they are going to be bound to, and then we'll look at how they're activated by receptor tyrosine kinases. Okay, we're not going to look in much detail at all at how Pi3-Kinase gamma enzymes are activated by beta-gamma subunits of heterotrimeric G proteins. Okay, right. So how is the regulation of these two different types of Pi3-Kinase type 1 enzymes so different? Well, it's because the regulatory subunit that the catalytic subunit is going to be bound to is going to be completely different in the case of the type 1As to the type 1Bs. Okay, so the type 1A Pi3-Kinase enzymes, which remember, these are characterized according to which catalytic subunit they have. So they're Pi3-Kinase enzymes which either have P110 alpha, P110 beta, or P110 delta as the catalytic subunit. Okay, they all have a certain type of regulatory subunit attached to them, which we'll talk about in a moment. And this regulatory subunit allows these Pi3-Kinase enzymes to be regulated by receptor tyrosine kinases. Okay, Pi3-Kinase gamma, which remember just means a Pi3-Kinase enzyme where the catalytic subunit is a P110 gamma protein, this is going to have totally different regulatory subunits bound to it. And as far as I'm aware, these regulatory subunits have not actually been crystallized yet. Okay, so we don't really know that much about the structure of P110 gamma's regulatory subunit. Okay, so we're not going to talk that much about the type 1Bs, we're going to focus on the type 1As. Okay, so let's now talk about the regulatory subunits then of both the type 1A Pi3-Kinase enzymes and the type 1B Pi3-Kinase enzymes. Okay, so if you are a type 1A Pi3-Kinase enzyme, okay, that remember means that your regulatory subunit is either P110 alpha, P110 beta, or P110 delta. Okay, so the regulatory subunit is one of P110 alpha, P110 beta, or P110 delta. Okay, the regulatory subunit that you are going to bind to is what's known as a P85 type regulatory subunit. Okay, now there are actually 5 different P85 type regulatory subunits, okay, and these are produced from 3 different genes. Okay, so I'll give you the names of these 3 different genes. So the 3 different genes which are going to produce these 5 different types of P85 type regulatory subunits are known as Pi3-Kinase, regulatory, that's the R, and then we've got regulatory 1, okay, then we've got the Pi3-Kinase regulatory 2, okay, and then we've got Pi3-Kinase regulatory 3. So these are the names of the 3 genes. Now, how are they overall going to produce 5 different proteins? Well, it's because some of these, well, specifically one of these genes is going to have multiple splice variants. Okay, so let me just remind you of the basic principle of splice variants. Okay, so let these 2 lines here represent a double-stranded piece of DNA. So it's a DNA duplex here. Okay, so let me just remind you of the central dogma of biology. Let's say that this portion that I'm outlining here, okay, which I'll highlight in blue here, let's say that this portion is a gene. Okay, for instance, maybe it's a Pi3-Kinase regulatory subunit type 1 gene. Okay, so this is our gene here. Now, one of the strands in the gene will be the strand that is actually used to produce the piece of mRNA. Okay, so let's say this portion that I'm highlighting in purple here, let's say that this is the strand that's actually going to be used to produce the mRNA, and the strand that's actually going to be used to produce the piece of mRNA is known as the coding strand. Okay, I'm just going to move this up so that I don't keep knocking it. Okay, right, the other strand that is complementary to the coding strand, which I'll now colour in green here, this is the non-coding strand of the DNA. Okay, right, now, what happens then is if you actually want to make the protein for this gene, what needs to happen is the Rna Polymerase 2 enzyme, which is often abbreviated to the RNA, P for polymerase, and then 2. Okay, so this is the Rna Polymerase 2 enzyme. This is going to come, it's going to work its way along the coding strand of the DNA, and it's going to synthesize a complementary piece of mRNA to the coding strand. Okay, so I'll colour this in turquoise here. Okay, this piece of mRNA is then going to work its way through a ribosome and be translated into protein. However, it's not ready to go through the ribosome yet. Okay, this piece of mRNA that we have got here is what's known as the pre-mRNA, or you can also call it the primary transcript. Okay, and the reason that this piece of RNA is not actually ready to go for a ribosome is that there are portions of this pre-mRNA that are not actually supposed to be translated. Okay, so let's say that these regions that I'm now boxing, let's say these are regions that are actually supposed to be translated. Okay, the portions that you actually want to go for a ribosome and be translated into a sequence of amino acids, these are called exons. Okay, meanwhile, the portions that are outside of these, the portions in between, okay, these four portions here, these are known as the introns. So the pre-mRNA has both exons and introns, okay, and in order to actually get this ready to go for a ribosome, you need to splice out the introns, you need to cut the introns out, and then you need to stick the exons back together. Okay, so you'll end up, therefore, with a much smaller piece of mRNA at the end of that process. Now, this process of cutting the introns out and then stitching the exons back together, this is known as splicing, okay, and it produces this shorter piece of mRNA, which is known as the mature mRNA, okay, which now just consists of the exons. This mature piece of mRNA will then be translated into a protein. Okay, right, in the case of some pieces of pre-mRNA, there is not just one way that you can splice your pre-mRNA. Some pre-mRNAs have multiple options for how you want to splice them, and these different options of how you want to splice them will result in the production of different mature mRNAs. Okay, so in the case of some pre-mRNAs, there are these different ways of splicing them, and depending on which method of splicing you choose, you will end up with a certain mature mRNA. And this means that if you've got concurrent pre-mRNAs which are all being spliced, different pre-mRNAs can go down different splicing routes, okay, and therefore can produce a range of different mature mRNAs, and these different mature mRNAs can then go on and be translated into different proteins. So an easy to understand example of how this can occur is, for instance, imagine that this final exon here is not actually a compulsory exon. Imagine that this is an optional exon. You can either include it or you don't have to include it. Now, if you include it, you'll end up with the full-length mature mRNA here. Okay, if you don't include it, you'll end up with a slightly shorter piece of mature mRNA. Okay, now these two different pieces of mature mRNA you can produce, one will produce a longer protein than the other. Okay, so that's how you can end up, well, that's a simple example of how you can end up with multiple proteins from the same gene. Okay, and these different proteins that you end up with that have been produced by the same gene are then called the different splice variants of that gene. Okay, so certain genes can produce multiple proteins by this variation in the mechanism of splicing. Okay, so that's important to understand, because this Pi3-Kinase R2 gene here is actually going to produce 3 different proteins. Okay, so it's going to produce a protein known as P50 alpha. Okay, it also produces a protein known as P55 alpha, and again the number just refers to how big the protein is. So this is the smallest one that this gene will produce. This is slightly bigger, and then we've got a bigger one still. You've then got P85 alpha. Okay, so those 3 proteins, P50 alpha, P55 alpha, and P85 alpha, they are all produced by the same gene, but they're different splice variants, okay? This one is a shorter version of this one, okay? It doesn't have some of the additional bits that the P85 alpha protein actually has. Okay, then for the Pi3-Kinase R2 gene, it's more simple. There is just one protein that you can then produce from this, and this is called the P85 beta protein. Okay, so notice all of the ones produced from the type 1 regulatory subunits gene have been labeled alpha, okay, the first letter of the Greek alphabet. The protein that is then produced from the type 2 regulatory subunits gene is labeled beta, okay? And then finally, the protein that's going to be produced by the Pi3-Kinase R2 gene is then going to be labeled gamma. Okay, so this is P55 gamma, which is the option for what you can create from this gene here. Okay, right, so these 5 proteins then, P50 alpha, P55 alpha, P55 gamma, P85 alpha, and P85 beta, these are the 5 different proteins that you can use as regulatory subunits for a type 1a Pi3-Kinase enzyme. Oops. So you can pair this with any of these catalytic subunits here. So each of these catalytic subunits, which create the 3 different types of type 1a Pi3-Kinase enzymes, can be paired with any of these 5 regulatory subunits. Okay, so that means that there are multiple different types of each of those Pi3-Kinase alpha, Pi3-Kinase beta, and Pi3-Kinase delta enzymes, because there are different regulatory subunits that you can have paired to the catalytic subunit, but this doesn't affect the naming because they are named after which catalytic subunit they have rather than which regulatory subunit they have. Okay, right, just to give you a complete picture, I'm going to talk about the regulatory subunits that you can now have for the type 1b Pi3-Kinase enzyme. Okay, so remember the type 1b Pi3-Kinase enzymes, they always had as their catalytic subunit P110 gamma. Now, the regulatory subunits are completely different for P110 gamma, okay? So, the 2 genes which code for the regulatory subunits that can be paired with P110 gamma are Pi3-Kinase R5. Okay, I don't know what quite happened to R4, okay, but then also there is the gene Pi3-Kinase R6, okay, so these are 2 genes, and each of them produces a single protein which can then be paired with P110 gamma to produce a full Pi3-Kinase enzyme. Okay, so Pi3-Kinase R5 produces a protein known as P101, okay, again labelled according to its size, and then Pi3-Kinase R6 produces a protein, and I shouldn't say equals, I should have an arrow. Pi3-Kinase R6 then produces a protein known as P84, okay? Right, so these are the 2 regulatory subunits that you can have paired with P110 gamma, and whilst these regulatory subunits up here, which are the P85 type regulatory subunits, these are all regulated by receptor tyrosine kinases, so as we'll see, they're going to interact with receptor tyrosine kinases, whereas these 2 regulatory subunits, these are going to interact with the beta-gamma subunits of heterotrimeric G proteins. Okay, right, so I think we'll call it there for this video, and in the next video what we'll do is we're going to continue our study of Pi3-Kinase enzymes. Specifically now we're going to focus on type 1a Pi3-Kinase enzymes. We're going to look at the structure of type 1a Pi3-Kinase enzymes, both the catalytic subunits and the regulatory subunits, and then what we're going to do is look at how they're going to be activated by receptor tyrosine kinases.